Hello and welcome friends. Welcome to this video on classification of enzymes. International Union of Biochemistry and Molecular Biology have classified enzymes into 7 classes as follows. Number 1, oxidoreductases. Number 2, transferases. Number 3, hydrolases. Number 4, ligases. Number 5, isomerases. Number 6, ligases. And number 7, translocases. Let's see them one by one. First, oxidoreductases. These enzymes catalyze electron transfer oxidation reduction reactions or redox reactions. In this example, there are two substrates A and B. A loses its reducing equivalents and B accepts those reducing equivalents. A undergoes oxidation and B undergoes reduction. Example, alcohol is oxidized to aldehyde and NAD undergoes reduction. Example of enzyme, alcohol dehydrogenases. If the reaction involves direct participation of oxygen, then enzymes are called as oxidases. And if the reaction involves the transfer of hydrogen, then the name of enzymes are dehydrogenases. Second, transferases. As the name suggests, these enzymes catalyze the transfer of a group other than hydrogen, such as amino group, carboxyl group, methyl group, phosphoryl group, etc., from one molecule to another. Yes, as you can see in this example, group R is transferred from substrate B to substrate A and a new product is formed. Example, phosphate group from ATP is transferred to hexose to form hexose 6-phosphate. Example of enzyme, hexokinase. Number 3, hydrolases. These enzymes catalyze the cleavage of carbon-oxygen, carbon-nitrogen, carbon-carbon and some other bonds with addition of water. Hydrolytic cleavage. Yes, the bond between A and B is hydrolyzed with the help of water and we get two hydrolyzed products. Example, acetylcholine is hydrolyzed to choline and acetate. Example of enzyme, acetylcholine esterase. Ligases. Now, these enzymes catalyze the cleavage of carbon-oxygen, carbon-carbon and carbon-nitrogen bonds by means other than hydrolysis or oxidation, giving rise to compounds with double bonds, or catalyze the reverse reaction by the addition of group to a double bond. That means these enzymes will catalyze the cleavage of these bonds and form double bonds, or they'll add group and remove the double bond. And in case where the reverse reaction is important, then synthase is used in the name. Let's see this now. We have this molecule, a substrate B, A, and two additional groups are there, X and Y. Okay. Now these groups would be removed by enzyme and a new product is formed with a double bond. The enzymes are called as ligases. So ligases will remove the group and form double bonds between the elements. Now if the reverse reaction occurs, then the name of enzyme is synthase, where the groups are added and double bonds are broken. Example, fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, one of the glycolytic enzyme, glycolytic step. Example, aldolase. Next, isomerases. These enzymes catalyze the intramolecular rearrangement in a molecule. Now, these could be structural or geometric changes. Example, A is isomerized to A dash. Example, again one of the glycolytic step. Glucose 6-phosphate is converted to fructose 6-phosphate. Name of enzyme, phosphohexoisomerase. Now the enzymes are called epimerases, isomerases or mutases depending on the type of isomerism involved. Next, ligases or synthetases. Now synthetases are different from synthases, which we have seen. These enzymes catalyze the joining of two molecules coupled with the hydrolysis of ATP. So ATP dependent synthesis, ATP hydrolysis will occur and that will supply energy for joining of two molecules. They may form carbon-oxygen, carbon-sulfur, carbon-nitrogen and carbon-carbon bonds. Yes, there are two molecules A and B. These are coupled to form AB molecule, a synthetic reaction catalyzed by enzyme, and that requires energy from ATP. So as you can see, ATP is hydrolyzed and supplies energy. Example, acetyl-CoA plus carbon dioxide are coupled, are joined to form a new molecule, malonyl-CoA, and ATP is hydrolyzed. Name of enzyme, acetyl-CoA carboxylase. Next class, translocases. Now these enzymes catalyze the movement of ions or molecules across membranes or their separation within membranes. Yeah. Example, a molecule B is present on side 1 of the membrane. Okay. Now this molecule will be translocated with the help of enzyme. Now as you can see now, the B has come on the side 2 of the membrane. Example of enzyme, sodium-potassium ATPase or sodium-potassium pump. So friends, let us quickly summarize the classification of enzymes. Enzymes are classified into 7 classes. First, oxidoreductases, transferases, hydrolases, liases, isomerases, ligases, and translocases. Oxidoreductases catalyze oxidation, reduction reactions. Transferases catalyze transfer of a group. Hydrolases catalyze hydrolytic cleavage. And liases catalyze non-hydrolytic cleavage to form double bonds or reverse reactions. Isomerases catalyze isomerization reactions. Ligases catalyze ATP dependent synthesis, and translocases translocation of ions or molecules. Let's see a very interesting mnemonic to remember this classification. A very familiar scenario in a medical setup, isn't it? So the mnemonic goes like this: Operation Theatre having loyal intern lady trainers. Operation O, oxidoreductases. Theatre T, transferases. Having H, hydrolases. loyal L, liases, intern I, isomerases, lady L, ligases, trainers T, translocases. So Operation Theatre having loyal, intern, lady, trainers, oxidoreductases, transferases, hydrolases, liases, isomerases, ligases and translocases. I am thankful to all these resources for making this video possible. Thank you friends. Thank you for watching this video. Be happy, healthy and stay safe. Thank you so much.